Hello everyone, myself Sandip, student of Namchi Government College, BSc Physics Department, is presenting you a short presentation on SEM and HRSEM, which was given to us by our respected teachers from Physics Department as our assignment for term paper 2. Before discussing my topic, I would like to express my special thanks and gratitude to my teachers who gave me a golden opportunity to do this wonderful project on the topic SEM and HRSEM, which also helped me in doing a lot of research and for came to know so many new things. Secondly, I would like to thank my friends who helped me in finishing this project in limited time. First, introduction part. Scanning electron microscope. A scanning electron microscope is a type of electron microscope that produces images of a simple sample by scanning the surface with a focused beam of electrons. The electrons interact with atoms in the sample, producing various signals that contains information about the surface topography and composition of the sample. High-resolving scanning electron microscope (HRSEM). The high-resolution scanning electron microscope is a versatile tool for nanotechnology and advanced material science. Moreover, opens up new dimensions in archaeology, biology, earth science, hydrology, and many other research areas. Diagram for scanning electron microscope. Here, F is the filament, C1 is the first condenser lens, C2 second condenser lens, S secondary coil, O objective lens, B backscatter electron detector, X X-ray detector, Y secondary electron detector, T image monitor. Principle. Scanning electron microscope. Unlike the transmission electron microscope, which uses transmitted electrons, the scanning electron microscope uses emitted electrons. The scanning electron microscope works on the principle of applying kinetic energy to produce signals on the interaction of the electrons. These electrons are secondary electrons, backscattered electrons, and diffracted backscattered electrons, which are used to view crystallized element and photons. Secondary and backscattered electrons are used to produce an image. High-resolution scanning electron microscope. The principle for HRSEM is same as that of scanning electron microscope. It is used for the better resolution. It is a versatile tool for the nanotechnology and other research areas. Now, theory. Scanning electron microscope. When it comes to scientific instruments, microscope let us see objects that are normally much too small for the human eye to see. The maximum magnification of optical microscope is limited by the wavelength of visible light. To get an image at a greater magnification, we need something with a shorter wavelength than visible light, such as electrons. Such a microscope that uses electrons for magnification is called SEM, that is, scanning electron microscope. Working of SEM. A beam of electrons is produced at the top of the microscope by an electron gun. The electron beam follows a vertical path through the microscope, which is held within a vacuum. The beam travels through electromagnetic field and lenses, which focus the beam towards the sample. Once the beam hits the sample, electrons and X-ray are ejected from the sample. Detectors collect these X-ray, backscattered electrons and secondary electrons and convert them into a signal that is sent to a screen. This produces the final image. Working for HRSEM. The electrons emitted from the cathode are accelerated by an electric field up to the required energy. The maximum electron energy in scanning electron microscope is usually 30 keV, which can be adjusted towards the smallest energies. After acceleration, the electrons are focused and scanned by magnetic lenses and finally produce the final image. It has fast imaging rate. Operating procedure. Signing the user log with the following information: username, data, time on, material being examined, including the specimens, types of work to be done. Second, loading of the samples. Use only one gloved hand when handling samples and holders. Never blow on or exhale on sample to dry them. Always make sure all screws are tight. Always ask if you have any questions. Third, bringing the microscope to operating conditions. Never turn the filament knob past its limit and always turn it slowly. Never move any of the stage controls past their limits. Never tilt a sample beyond the specified limits indicated on the specimen chamber door. Fourth, alignment of the objective aperture. Increase the magnification to well in excess of the level you wish to image at. Adjust the position of the objective aperture. Only adjust one direction at a time and adjust them until the movement of the images stop in that direction. Fifth, removing a sample. Never remove a sample with the voltage or filament on. Always wear a glove on one hand when manipulating samples and holders. Using that one gloved hand, push the large circular plastic plate on the sample exchange rod to the end of its travel. Using your one gloved hand, unscrew the sample holder. Sixth, shutdown procedure. Be sure that all the samples are removed from the microscope. Turn off the monitors on the compact and cabin units. Sign off on the user log. Remove all the samples and clean up film, etc. Turn off the room lights and shut door on the way out. Results. Results for SEM. A scanning electron microscope projects and scans a focused stream of electrons over a surface to create an image. The main component of SEM includes source of electrons, column down which electrons travel with electromagnetic lenses, electron detector, sample chamber, computer and display to view the images. Results for HRSEM. A type of electron microscope that produces images of a sample by scanning it with a high focused beam of electrons. The spatial resolution of SEM is limited by at least three factors: the diameter of the electron probe, the size and shape of the beam, and the position statistics of the detected signal. With the state-of-the-art electron optics, resolution of the order of a nanometer are now possible. Applications. Application of SEM: material science, forensic investigations, medical science, identifying diseases and viruses, and biological science. Applications of HRSEM. HRSEM is used to visualize the atomic structure of objects under study. It is used for measuring local variations in lattice parameters. It is a technique for obtaining high-resolution images of biological and non-biological specimens. It is used in biomedical research. So lastly, future scope. It is a very fine future scope as the humans have tried continuously to improve their abilities of seeing micro things and to reveal bacteria, viruses, etc. from developing microscope. There are lots of workshops being held to bring together experts from various fields to identify new science technologies that will be enabled by advances in high-resolution electron microscopes. Thank you.